At the start of the school year, spotted lanternflies had infested our campus. The spotted lanternfly is an invasive species that came from Asia and is plaguing a large portion of northeastern United States. Based on our initial research, it was hypothesized that neem oil would be the most effective, non-toxic treatments for eliminating spotted lanternflies.
As seen here, lanternfly traps were set around the school campus to collect egg masses for testing. Trap construction was based on work by Lewis and colleagues at the USDA. Spotted lanternflies are known to prefer the tree of heaven for laying eggs, but there are no tree of heavens on our campus. This conduit tree proved to be effective in collecting spotted lanternfly egg masses.
Once sufficient egg masses were collected, individual egg masses were divided into small samples and non-toxic treatments were developed. Solutions of neem oil, eucalyptus oil, lavender oil, and vinegar were tested against a control of distilled water. 6 sprays of each solution were applied to each sample. Treatments were done in quadruplicates. Samples were placed in a refrigerator to simulate winter.
Meanwhile, live spotted lanternflies were tested with the same treatments in quadruplicate. Live flies were placed in flasks with a piece of apple to assure that flies would not die from starvation or dehydration. Treatments were sprayed onto adult spotted lanternflies. Both female and male spotted lanternflies were tested to assure there was no difference between the two sexes.
For the live spotted lanternfly samples, vinegar proved to be the most effective treatment, with 75% of the samples dying within the first 22 hours of treatment. Eucalyptus was the second most effective treatment, and neem oil was the least effective, with 3 of the samples living as long as the control. As proof that starvation or dehydration should not impact results, all 4 control samples lasted the longest, with the last live spotted lanternfly dying around 65 hours of captivity.
After 23 days in refrigeration, the egg mass samples were transferred to 15 degrees Celsius incubators to mimic the onset of spring. Data was collected for 33 days daily, and the experiment ended at the end of our semester. For the egg masses, none matured to live lanternflies. They did progress towards hatching. Similar to the live flies, the vinegar treatment seems to work the best, having resulted in the most inhibition of the egg mass development. Lavender oil also proved effective. Of the 4 treatments, 75% of the eggs exhibited only slight progress towards hatching, and 1 egg mass exhibited little to no progress. Neem oil and eucalyptus oil were the least effective. The majority of neem oil treatments showed major progress towards hatching. The majority of the eucalyptus oil treatments showed moderate progress towards hatching.
In conclusion, our hypothesis that neem oil would be the most effective non-toxic treatment to kill spotted lanternflies was not proven, but we did show that vinegar and lavender oil were both effective treatments to deter both the live flies and the egg masses.